Good morning, everybody, and thank you for joining us for our W.E.S.T webinar here in the month of April. Wanted to welcome everyone for joining us. Thanks for making it. Today is April 28th of the year 2021. And today we will be having our guest speaker, Dr. Gaurav Sahay, present his session on enhancing endosomal escape and applications to the extrahepatic mRNA delivery. A little bit of housekeeping before we start, of course, as always. Wanted to introduce your W.E.S.T regional team, starting with the Southwest region. Our regional sales manager based out of the San Diego area is Kim Killian. We also have our field application scientist based out of the, Southwest area as well. Closer to Irvine, we have Viet Nguyen. Here in the Northwest region, we have our field application scientist, Ian Viamanya. He's based out of the San Francisco Bay Area. And of course, I am the regional sales director, Angel Galarza, and I'm also based out of the West region and based out of the San Francisco Bay Area. Reminder, of course, we do have these quarterly events, and actually these happen every month. We have them set up for you guys to join us. Our upcoming event, of course, in the month of May, is we have our esteemed colleague, Dr. John Lewis, out of Entos Pharmaceuticals. That will be on May 26th. We also have, of course, one of these similar webinars here in the month of June with Dr. Justin Rickner from the University of Illinois. And of course, as you may know, that we have our monthly journal clubs, which are the last Fridays of every month. And we are starting a new quarterly event called the Lunch and Learn, which we'll have once a month. Thank you for joining us for all of these, and hopefully we'll see you in the future as well. Last thing, of course, if you miss any of these, please know that all of our webinars are on YouTube, as well as some of our journal clubs and Lunch and Learn. So you can go to our Precision Nanosystems channel and ask or to find these particular webinars or journal clubs. Just type in Precision Nanosystems and you'll find this. So I'll hand it over to our guest speaker, Gaurav. On you. Thank you for joining us.
Thank you so much for the kind invitation, Angel, and the Precision Nano System team. I'm very grateful for this opportunity. So what I wanted to do today was to talk about extrahepatic gene delivery and, discuss some of the work that we are doing and where the field is sort of heading into. And then finally, we might have some time to discuss and have some Q&A. So what I'll talk to you today is I'll just give you an introduction about RNA delivery, then talk about the endosomal escape aspect and intracellular delivery of mRNA, extrahepatic applications, and extrahepatic targets. So, now it's, we used to talk about mRNA pre-COVID and how it has the potential of moving very quickly in terms of like gene, as products. And now we have real world sort of examples. So it's a very surreal, thing for all of us in the field as this moves forward. So the advantages, as we now know, are there is no risk of genomic integration with mRNA. It doesn't need to enter the cells. It's highly efficient. The effects can be rapid, and it's transient and regulatable. So it's very useful then for a vaccine-based platform. The challenges are that the mRNA is very negatively charged. It's unable to enter cells, and it's highly susceptible to degradation. The lipid nanoparticles are, because this is a charged, negatively charged molecule, it requires something to carry it inside the cells, and it also gets broken apart as well. So, um, what the it requires an enabling technology, and the enabling technology in this case is a lipid-based nanoparticle, which can encapsulate RNA. Um, and it's composed of multiple components. The ionizable cationic lipid that interacts with an RNA, encapsulates it, a PEG lipid that covers the surface, a messenger RNA, and structural lipids. And how you make it is you put these in a microfluidic mixer, most likely from Precision in the lab, which makes a very good 80 to 100 nanometer particle. And the thing with this is that it can package and protect nucleic acids. It can have a large cargo capacity. So these are very modular in nature. You can put mRNA, SI, or genome editing components. Some of them are biodegradable. There is capacity to redose. These are modular in nature, and scalability. Again, pre-COVID, there was always like the question of scale, but I think that's now being handled. A lot of us have already received vaccines that are made of lipid nanoparticles. So I think that's something very phenomenal that's happening to the field. And then finally, there is, the first FDA approved nanoparticle formulation was on Patro, that got FDA approved for sRNA delivery. And these are perhaps the main actors. They're the ionizable lipids. The MC3 is the first one that got approved for sRNA delivery. There's another category of lipidoids. There are these next generation lipids, the Acutus lipid, Moderna lipids. And I've not put the structures of the newer ones, which have been injected in a few of us in here. But, they're basically from the same next generation of series. So, these are the first product. So Onpattro was the first product with a lipid nanoparticle for sRNA delivery. But now we have mRNA vaccines. And again, this has been one of the surreal moments, which we, from just an year and a half ago, when the pandemic hit, to where we are now. In this case, you have a spike, coronavirus, which has a spike protein. So very quickly, an mRNA against the spike protein was made with a 2p modification. So it is in this pre-fusion complex. And so both Moderna and Pfizer BioNTech Acutus have this sort of like mRNA with this 2p modification in it. Both have lipid nanoparticles, and lo and behold, have very similar results to prove from, where you saw in a phase 3 clinical trial 95% efficacy. Now it's been given to a lot of people, and again, like the impact has been immense currently, but I think also for the field. So if we are able to now scale these lipid nanoparticles to a level of billions, tons of material, so then you can say that, like, if you have a new LNP based system that can reach to the lung, you can basically deliver genetic cargoes to the lung for diseases like CF. You can actually supply these for the rest of the life of a rare disease patient, because the subpopulation is less, if it's not like a one shot and done kind of thing. So the field is moving from what I call a renaissance period to a full-blown industrial revolution as it goes. And there are a lot of opportunities here for students who might be listening, to work in this field and get a lot of jobs as well. But the next phase, we think, is going towards the extrahepatic delivery. So from meaning thereby that it's moving from, delivery into different organs than just the liver, because that has been the primary target of these LNPs. So to the heart or the lung, gut, there is some evidence that, these have been delivered for placental targeting as well. Um, and then my lab works in the space of eye and the lung for delivery of lipid nanoparticles to deliver genes. So in my talk, I'll be talking about LNPs that are composed of DLN MC3 DMA, DSPC, DMG-PEG, again for stability, and cholesterol. And how does the lipid nanoparticle work? So an LNP, if injected IV, the PEG lipid, which is on the surface, basically falls off. ApoE, a protein, binds to it, takes it through the LDL receptor, from where some dissociations happen, and the ionizable lipid, which now carries a positive charge, interacts with an endosomal membrane, leads to cytosolic sort of delivery, and that causes either gene silencing or gene delivery. So the escape with MC3 was basically found to be around less than 2%. With mRNA and newer lipids, it has improved to 10 to 15% as well. But still, this remains a huge problem. And the majority of the particles are also recycled out. So when I was a postdoc, we actually figured out that the recycling process was dependent on Niemann-Pick type C1 protein, which is a late endosomal protein and is used for cholesterol transport. So we started looking and at that time working with Moderna to understand if cholesterol has a role to play in the trafficking of these particles inside the cells. So we got a bunch of naturally occurring cholesterol analogs and made different groups within them, and figured out that, first we made different, it had the same MC3 LNP, but different cholesterol analog replacing cholesterol per se. So what we found was that there was a group of lipids that actually had a really good nanoparticle encapsulation and had like a 20 to a 30-fold improvement in gene transfection. These were cholesterol analogs made of beta cytosterol, which has this C24 alkyl, which is a C24 alkyl derivative. And from there, we started to look into how does this exactly work. And it took us a long time to figure out why this was working. And we also got a gift of Lipid 9 from Moderna at that time. And as you can see, the encapsulation efficiency of these particles are the same. The hydrodynamic diameter sort of is a little larger for cytosterol and has a low polydispersity. But what was intriguing to us was to find that these had actually like these polyhedral structures. So if you add a cholesterol analog, it has these polyhedral type of structure with beta cytosterol, whereas with just plain cholesterol, you see this multilamellar structure. So the shape of the material was becoming polyhedral. The MC3 or lipid 9 independent of that, and internal structure, as when we looked through Sachs analysis, was not much different. So once you then transfect, we said, let's see how good lipid 9 transfects. And as you can see here, the transfection really blew apart. With this system, you could do like delivery of gene editing agents, and that was pretty high. You could also do gene silencing with siRNA. And amongst different cell types, you'll see different efficacy. So depending on how much uptake is happening, you see a difference in gene delivery. So then what we did next was we took different analogs of cholesterol and started to look into how these exactly look like under a cryo-EM. And a postdoc of mine, Julia Egres, actually did this work, who basically figured out that there were some differences amongst these analogs that we were observing, when you look at the structure. But with the materials that work really, really well, for transfection, cytosterol or stigmastanol, you see these kind of polyhedral shapes happening because of changing that with an analog. So when you then look under a multiple particle tracking, single particle tracking method, we actually were able to observe that like these particles basically are bumping around an endosomal vesicle and sketching that. Whereas the ELNPs are the ones which have cytosterol in them. So these are the enhanced LNPs. So LNPs, the usual traditional LNPs, the conventional ones. With the ELNPs, you see this, linearity that it gets. So which means that it perhaps is going through certain pathways that are much more amenable for escape. It doesn't really show escape, but it does show that it's in an environment which is at an intracellular level, which is very different from an LNP. So then we wanted to, know if this was exactly the case. Are we seeing a higher endosomal escape? And what we then did, like there is some beautiful work that came in from different labs that showed that if you basically use galectin, which is a cytosolic molecule, if there is a breakage in an endosome, this galectin 8 binds to that endosome, and this could be used as a marker for endosomal escape. So, so galectin 8 or 9 are sitting in the endosome. Once it breaks, it gets trapped. It you know goes into the surface of the endosomal membrane, and that can be a good sort of a way for us to visualize whether escape is happening or not. So then what we did, this was done by Marco Herrera in my lab, grad student, along with David Kim, who who made these cell lines uh, where you can see galectin 8 in the cytosol when it's in an untreated form. You add LNP, which has cholesterol in it, you see these rare events again, right? Like the escape is so low. So all those endosomes that are helping in escape, those are, showing this punctate structures. But when you add the one that has cytosterol, you start to see this massive endosomal sort of leakage happening and escape happening. That confirms that the addition of these cytosterols are changing the structure in such a way that is leading the particles to somehow manage to have a better escape than the conventional systems. Okay, so next, what we wanted to do was to use this strategy to deliver different mRNA. And during the same time, when COVID hit, we said, okay, there's a lot of activity going on in the vaccine space. Can we use mRNA as a strategy for therapeutics? So we devised in a way in which we could actually deliver the soluble part of ACE2. So the viral the coronavirus enters through this ACE2 receptor in the lung, which also is an enzyme which is responsible for angiogenesis. So when we added, so we thought this is a transmembrane protein, but it also has some sort of soluble character. So some of it is also found in the soluble form. So what we thought was that we'll deliver the soluble form of ACE2, and that could basically capture the virus, and we could give it as an inhalation or also an IV injection. And through the IV injection, the liver will become a factory for the production of soluble ACE2. For the lung, we will have production of soluble ACE2 directly in the lung and see which works better. So this is the endogenous ACE2, and this is the variant form, which doesn't have the transmembrane domain, but we did add some tags so it becomes easier for us. So again, this was when, during the pandemic, when it had just started, we moved very quickly in this area as well. So what we found was that ELNPs were able to produce a lot of soluble ACE2 in the culture media, as detected by Western blot. And then also in lung cells, we saw a lot of soluble ACE2 being formed. The ELNPs, if you do nasal instillation, you could get lung transfection. So this is just a luciferase as reporter. You start to see expression into the lungs. And when you then give soluble ACE2, again through nasal instillation or intratracheal instillation, you start to see soluble ACE2 that takes into form, and you can actually measure in the bronchial alveolar lavage the formation of soluble ACE2. So next, again, this took us a long time during that time to actually also, make a pseudovirus against the spike pseudovirus, which had a luciferase in it. And as you can see here, in cells that have overexpressed ACE2, you see a transfection or transduction using the spike pseudovirus. Once you add the soluble ACE2, formed by the lipid nanoparticle delivered mRNA, you start to see around 95% inhibition. As a positive control, we actually took VSV, which hits all the cells independent of ACE2, and you see there this technology does not work. So now at this point, now we are collaborating with some folks to push this in vivo. And again, this was done by David Kim in my lab and in collaboration with Andy Mukherjee, who grad student and postdoc respectively, and a couple of other folks that I'll talk about in the end who helped as well. So I think this was we wanted to actually move and at an academic level and see the utilization of mRNA as a therapeutic, and hopefully as we move in vivo, we see some efficacy there as well. One other application pre-COVID we were working on is to deliver CFTR mRNA. And CF is a disease of the lung. The absence of CFTR causes dysregulation of chloride secretion, which thickens the mucus. So CF patients have a very thick mucus. And so one of the things then is if we could deliver CFTR gene, and it has been a long-standing goal as a therapy for these patients. So CFTR, there are around 1,700 known mutations, and there are a couple of drugs that have worked really well and have started to resolve the problem. But if we could deliver in cell types CFTR mRNA or now gene editing components, it could help patients independent of their genotype. And it could, with mRNA, independent of genotype, we could basically give the same therapy. So what we did was we took the lipid nanoparticles, made a CFTR mRNA, delivered it inside the cells. And how you measure CFTR function, the first way to see if it's functional or not is, giving the particles in the nasal cavity. And in the nasal cavity, then, the chloride function is then looked by, putting some electrodes in. And this is done clinically as well. So we have a clinician, Dr. Kevin McDonald, who we work with. And what we found was that we were able to get around a 40% response in CFTR, which is quite good because some of the drugs like ivacaftor give like 40% responses. So this is the first step. But, the next step would be if, we can now deliver the CFTR into the lung and see that we have we can first cross a thick mucus because CF mouse doesn't have any mucus, so you have to make different use of different models to find a way to deliver the gene across the mucus, deliver the gene, and then have a functional assay that basically within the lung you can have an effect. But this is like the first proof of concept that we had published. Only recently, we also, ENaC is another target which people are trying to use an siRNA against, because what happens in CFTR knockout condition, ENaC becomes hyperactive. This pushes the sodium out of the mucus, and wherever sodium travels, water travels. So the mucus becomes very thin or very thick. So a lot of people have this theory that if you can silence ENaC, you can actually restore function. So, folks are delivering siRNA as a technology for that. But we thought, why not use mRNA to inhibit a channel as well? So what Andy Mukherjee did in my lab was that he made an alpha mutant mRNA, and that basically the ion channel is made of alpha, beta, gamma trimers, and you need alpha to have a functional channel. So if you make a mutant alpha subchannel, then it basically would inhibit it. So that's what we show in our recent paper in Science Advances, that you can actually use at the alpha mutant ENaC and then start to see changes in conductance in the nasal epithelia. And then our goal then is that you can combine ENaC with CFTR mRNA and see if it has a better function when given in combination. So hopefully, we could not just give back a gene, but also can inhibit a gene using the same mRNA-based approaches. So one of the other things which we are also trying to do is to nebulize these particles, so that rather than instillation, you could just nebulize, and it's much more better for the patient. It has large dosage area, and the delivery is through constricted airways. The challenges are that the current technology is a vibrating mesh, so it leads to a lot of dissociation of the LNPs. So we are currently working into developing different ways and methods and formulations. If you make these particles a lot stable, then they might cross through, but then they won't work because they'll once inside the cells, they'll not be functional anymore. So we are coming up with the ways and making progress in terms of nebulizing these particles as well. The one of the other application is towards the eye. So using the same, like I said, extrahepatic as our goal, one we move towards the lung, and the other area that we moved towards was the eye. And the eye, has the retina that provides um, which provides visual acuity, and it's composed of different cell types. The most importance of those in terms of like getting inherited retinal diseases is the RPE. That's a phagocytic sort of cell that basically takes over whenever the outer segments of photoreceptors are done. It basically clears that, and new outer segments are formed. So mutations in the RPE leads to degeneration of the retina that can cause blindness. And the other area is the photoreceptors, because that is another the main site where which gives again visual acuity. And there is degeneration because of mutations in these photoreceptors as well. So these two cell types are important targets for us to deliver genes. There is one drug, Luxturna, that has already been approved. The way Luxturna is given is in an AAV vector. You give it in the back of the eye subretinally, and for long-term RPE 65, the gene is restored. So that has shown some promise, but the challenge again with AAVs is that you can't redose them, or it's a one-time shot, and large genes, especially AAVs are unable to package them. So in that regard, we need new delivery vectors, especially for large genes for the eye. So there are different routes of administration for the eye. The first one is the subretinal, because a surgeon comes in, injects subretinally. It's a local delivery and has been the way to deliver a lot of like gene editing components. But the challenge is that it doesn't cover the whole retina. It's only the subtype of the retina that it covers. And again, you require a couple of clinicians to be together to do this procedure, and it can be only done one time. The other idea is to deliver a drug, and this is done very regularly in the ophthalmology field, um, as even as an outpatient, I think, service, where you could give a drug intravitreally. The challenges with lipid nanoparticles or AVs that there is this huge mesh of hydrolunic acid and different barriers, and a lot of the material then just gets stuck in this region of the vitreous and doesn't cross. So there is an inner limiting membrane, and then you have like around 100 micrometer of different cell types to cross to reach to the back of the eye. So, it's not a easy thing to do, and a lot of people are, have worked with AAVs to come up with a way, because then you can give the drug multiple times. So what we did here was we first tried to look at different ionizable lipids and see what cell types do we hit. Rene Reyes and like Sid Patel from my lab actually did a lot of these studies where we injected subretinally an mRNA. And what we find is that you see a lot of the RNA transfect just the RPE. So if you want something to be delivered to the photoreceptors, you might not be able to, and that too, just subretinally, you can go to the back of the eye and deliver genes with like different ionizable lipids, out of which MC3 and KC2, these guys, which are, perform really well. So, for some diseases, this might be good, but we also want to reach to the photoreceptors because the large gene mutations are happening in the photoreceptors as well. And once you inject these particles subretinally, you do see a pan-retinal expression, which is again different from usually what AVs do. And we also see some Mueller glia that also shows, which is a macrophage-based cells in the retina, that also shows some expression, and that might be interesting for certain applications, which are towards like regeneration of the retina. So what we are trying to do now is to come up with a host of different ways and put a barcoding on these lipid nanoparticles, inject IV, and then figure out what are the structural features of the LNPs that can reach to the back of the eye, and also then develop ways that we could do multiple injections in the eye as well, and see if we can come up with new cargoes and new delivery mechanisms that can transfect the photoreceptors. The other thing we are doing is to do a lot of you know um, uh, the studies don't translate from the eye into a non-human primate. So we are developing like an ex vivo model of a non-human primate. This is a non-human primate eye. Inject, subretinally, different particles, and then have a barcoding approach to see which particle actually goes to the back of the back of the eye. So and we are validating this process and figuring out which cell types can are amenable in a non-human primate, as well as we'll be trying in live non-human primate as well some of the studies to identify which particles actually cross to the back of the eye. So the challenge with the eye is if you give the particles intravitreally, they do not go to the back of the eye, right? So all the data I showed you before, it's subretinal. You cannot do a repeat injection after subretinal. It's good if you have a one-time shot like an AAV, or for genome editing, it still works. But if we had a way to give through the vitreous cavity, it becomes much more easier. With non-viral vectors, again, if you want to do a repeat dosing, that becomes also very possible. But we think that it's not just possible with making different formulations of LNPs. We need some ways of targeting to the back of the eye as well. So using phase display, we have identified some peptides. Again, Marco Herrera in my lab has done that study, and we have a peptide that actually hits the photoreceptors really well. And as you can see, the the outer nuclear layer lights up, and it's pretty rare to see. And right now we are trying to use the nanoparticles, and we have interesting data coming up there as well, in terms of like gene delivery to photoreceptors, using some of the strategies that we are developing in the lab. But I think we'll maybe next time I'll be talking about them. So what does my lab do? We make lipid nanoparticles. We try to understand how do they work, what are the trafficking mechanisms through which the process of endocytosis happens, Then we are developing inhalation-based products as well. We move towards the decoy ACE2 or the soluble ACE2, as they say. We are also working in the CFTR space and trying to develop materials that can cross the thick mucus and reach the specific genes. It could be, ocular delivery as well. We are very much invested in ocular delivery and delivery to the back of the eye. And through systemic delivery, we are trying to reach other target organs as well. And currently, we are developing this for mRNA delivery and for gene editing kind of scenario. But I think the future is, very bright in the field. I think there'll be a lot of changes and a lot of rapidity through which things would move now. It was already going in that direction, but as I said, it's moving from a renaissance to a full-blown industrial revolution. Most of the IV injections goes to the liver, but through muscle now we know that we can hit certain immune cells. People are working in the lung space, kidney, gut, and as I showed you, intravitreal injections or to the lung. There are also evidence of direct injection in the brain. People had published before that if you directly inject, you see some expression, but crossing the blood-brain barrier still remains a challenge. But I think that's something people again would look with great interest as well. But you see some evidence, in publications on these different organs that I have depicted. And I think, there are new things like SORT that go to the lung with a new way of making the lipid nanoparticles. Intraperitoneally, it hits the macrophages. There were paper, going for cancer-based immunotherapy based on that. There is a lot of interest in DNA barcodes to generate a lot of lipid nanoparticles and then identify quickly in vivo how to screen for these particles. And of course, I think there's a lot of new material need. So it's not a saturated field at all at this point. In my view, there is a lot of ways people could make different materials and test and look at how exactly they work. Because I think that's also an important part. We need to understand how these things work and then develop much better and better versions. If we just continue to not understand how they work, we'll not know if something fails, why, there will not be a learning curve there. So I think that is an important fund. So as this moves further, fundamental studies remain very important as well. So finally, I'd like to thank, my current and former students, postdocs who have done a lot of work. Sid Patel, who's at Senda, who did the cholesterol analog story. David Marco, Anthony, who's done a lot in terms of the pseudovirus studies. Dr. Rayals, who's working in the eye, and Dr. Yulia and Mohit, who are working on the LNP side. So, and also my collaborators for CF, Kelvin McDonald, Mark Panisi, and, briefly, we also work with Moderna for the cholesterol analog story. We have been, funding through the CF and NIH. And with that, I thought, I'll finish a little bit ahead so that we get a long time for a discussion.
Awesome. Thanks so much, Gaurav. Really appreciate that talk. Angel, now just to close us out.
Gaurav, thank you so much for making it. Really appreciate it. As always, I know that you're slammed, but you're always one of our champions, and I really appreciate you doing this for us multiple times now. I appreciate you being a champion of the technology, and for being there and making all these advancements in lipid nanoparticle, the technology in general, and just these next steps for the future for these particular medicines in general.
Thank you so much.
Thank you again, and we'll see you next time, everybody. Thank you, Gaurav.
Bye. Bye.
Take care.